Molecular biologist Elizabeth Blackburn, one of Time magazine's 100 most influential people in the world in 2007, made headlines in 2004 when she was dismissed from the President's Council on Bioethics after objecting to the Council's call for a moratorium on stem cell research and protesting the suppression of relevant scientific evidence from its final report. But it is Blackburn's groundbreaking work on telomeric DNA, which launched the field of telomere research, that will have the more profound and long-lasting effect on science and society. In June, we hosted Catherine Brady, who told the story of Elizabeth Blackburn's life and work and the emergence of a new field of scientific research on the specialized ends of chromosomes and the telomerase enzyme that extends them. Today, we welcome Elizabeth Blackburn herself to our Google San Francisco office.
Thank you, Shannon. Thank you. It's a really great pleasure to be here and to have this opportunity to talk to you, and particularly to talk to you about the science that I do. I know Kate Brady talked to you about the context in which the science was done, and she talked very much about the issue of women in science. But today I really want to tell you about the science. But as you'll see, it's certainly very much about people. Now, where does the research that we do come from? I thought I'd give you a couple of slides just to kind of put it in some context. Here we are in San Francisco. This is the Mission Bay campus, still being built in certain ways at UCSF. Here we are somewhere up here in San Francisco, not very far away. And I work in this big building here, and if you look in the atrium, you see a lot of layers of different floors and different labs, and inside there are people who are, in this case, spoofing around a little, and this is where we do science in cluttered kind of laboratory setups. But we sometimes go outside and have barbecues. Here we are in front of the Mission Bay building where I work. And I want to introduce you, though, to the most important slide that I think I can give, and that's the slide that shows the folks in my research group. And so this is a fairly typical kind of research group in an academic setting, and there are, as you can see, a bunch of folks, and they do different things. And so, for example, Dana and Kyle and Josh are technicians who run experiments and do certain lab management things. And then there are people at different levels of training. So, for example, I have postdoctoral fellows, and these are people who have their PhD and do research for a few years before launching their own independent careers one way or another. And I've highlighted Zhe and Li Fang and Shang, Imke and Tanya. And then we have students of different stages of getting their PhDs, and some will be entering the program of PhDs, some will be in the middle, some will be just finished. So we also have clinical fellows. These are people who have MDs and PhDs. This is the case for Jason and Brad, shown here, and they are continuing their fellowship, further training after their MD and PhD in research, so they can be physician scientists in one form or another. So I just wanted to put a human face on the kind of people who do the research that I'll be telling you about. And they really are the people I want to acknowledge, because they do much of the work that I'll be condensing for you in today's talk. So let's put the talk into a general context. This is a pretty common sort of sequence that we're all familiar with, and it raises the question, how do we age? Now, I come as a molecular biologist and a cell biologist, and to some degree someone trained in genetics. All these fields merge together in modern biology. But this long-standing question has been around for a long time, and I actually came to it relatively recently, and I'll tell you as I go through the talk how that happened. But first of all, let's think about this question of aging, and one thing is very clear: it's multifaceted. You can think of it not only as biologically having all sorts of different facets, but socially, economically, philosophically. There are many, many facets to aging. Now, I'm going to focus obviously on the biological facets, although they've been very interesting to think about what are the bigger social contexts of aging, and particularly as we really learn more and more about its underlying nature. One aspect which is relevant to the kind of science that we do, and I'll tell you about, is the facet of aging which is increased susceptibility to diseases of aging. And another question you can ask, well, how much of this is environmental, and how much is genetic? And that's a long-standing question, and that's what I'm going to be telling you about, how we got into looking at these questions, kind of unexpectedly. Okay, so we all love a successful aging story, and this is one of my favorite pictures. This is the late Dr. Marguerite Vogt, and she's shown here at the Salk Institute, and she was probably well over 90 when this picture was taken. She died, in her 90s, but she could come to the lab and be a productive scientist and just have a marvelous time into her 90s. So this is the kind of example of successful aging that, we all hope to see. Now, as a biologist, what we want to do is ask, well, what goes on? And one kind of question you can ask is, let's imagine of the 100 million cells in her body, let's look inside a cell, right? And if we look right at the heart of the cell, what we'll see is the genetic material. And these blue sausage-like things are the chromosomes. And the red dots at the end are a molecular probe that is picking up the telomeric DNA. And that's fluorescing red because the telomeric DNA is found at ends of every chromosome. So I'm going to tell you now about that. And I'm going to use various kinds of images along the way to kind of, I hope, make some of the concepts a little bit clearer. So forgive me, I'm going to be mixing some metaphors, but I really enjoy visuals and imaging, and so I'm going to be showing you a bunch of different kinds of images as I go along to try to illustrate the concepts. So the simple statement, which won't mean a whole lot at this stage because I haven't explained this yet, but essentially genetic information is protected by telomeres and telomerase. Now I'll introduce these. And I'm going to organize this talk in the following way. I'm going to talk about telomeres and telomerase and talk about, first of all, their discovery, and then how they get implicated in human health and disease, and particularly in very recent work, how we've been, with our collaborators, been able to relate life factors and cell aging, bringing together two rather unexpected kinds of things. And that's the fun and excitement of science, is when really unexpected things come together and new insights start to emerge. Okay, so telomeres simply cap off the ends of chromosomes. So let's have a little metaphor here. So if you bought a very expensive pair of shoes online, such as the pair that I would never buy, but shown here, what you get for your outlay is at the end of the shoelaces, you'll see that there's a nice little protective tip. And this actually has a special word in the dictionary. It even is called an aglet. That's a special definition. And it's a tag or a sheath, like at the end of a lace, which in this case helps it get through the eyelet holes. But it also helps the ends from fraying away. So we can think of the chromosome. And I showed you a picture of a whole lot of chromosomes, the entirety of our chromosomes, those blue sausages before. But each chromosome is a linear entity, and it's got all the genes arrayed along it. And so we can think of it as this shoelace with the protective ends being the aglets. Those are the telomeres. And I can add a little bit more technical stuff, which I will explain a little bit more in a moment. But it's got DNA and proteins and so forth, and I'll tell you more in a moment. And we can see that what it can do is prevent this from happening. So my graduate student, Chris Smith, generously donated his sneaker shoelace for this demonstration of what can happen if you don't have a functional aglet at the end of your shoelace. So the analogy with the chromosome end is that it frays away, and it's actually a pretty good metaphor. Okay, why would ends fray away? There's been this problem since the understanding of how DNA was replicated, that a linear DNA, and the chromosome has a linear DNA running from one end to the other of the chromosome, that a chromosome won't be able to be replicated by the normal enzymes that normally can completely replicate all of the genetic material. But there's a glitch in that machinery, and I won't go into the details, but it's well studied, and it doesn't allow the very ends to get copied. So as a cell multiplies, each time it multiplies, of course it has to replicate its DNA, and then replicate its DNA again the next time the cell multiplies, and on and on and on. So those are called cell divisions, because the cells divide in two each time. And so what happens is that the DNA left unopposed would be predicted to have this sorry fate, which would get shorter and shorter and shorter. And there was even a prediction that perhaps this would even lead to cells eventually, running out of the ability to keep multiplying. This was a theoretical prediction made in the 70s, and nobody knew what the molecular structure was at the ends of chromosomes, but there was just this idea based on the DNA replication properties. And so what I was able to do was to study the molecular nature of the ends of chromosomes. So I'm going to show it to you in sort of schematic and not overly technical way, but imagine, so here's a chromosome. So if we zoomed in and looked at the end, we would see the very last portion is made up of little repeated motifs of the building blocks of DNA. One of them happens to be G, and it's got plenty of G's in it. There's the four building blocks in DNA, A, G, C, and T, the familiar blocks. And this one happens to be G-rich for reasons that are probably lost in the mists of time. But this DNA is this hundreds or thousands of these little repeated units over and over and over. Now, we're familiar with DNA for coding for proteins. This DNA doesn't do that. What it does is that the details of the DNA, because it's made of this repeating sequence, make a sort of geometry that acts as a kind of molecular scaffold that attracts a lot of proteins. And these are specialized proteins. They come in and they kind of sheath the end of the chromosome. Okay, so you need that to be a functional telomere. There's two ways a telomere won't be functional. One is if the array of repeats gets too short. And I've just shown you that there's a threat that chromosomes are under all the time. If they keep replicating without some compensatory mechanism, they will get shorter and shorter. Unless something else happens and comes to the rescue, that'll be telomerase, as I'll describe, or if somehow you disrupt the proteins off the end. But the consequences are dire, because when this happens, all sorts of alarm bells go off in cells. They don't like this. They don't like the fraying away of their chromosome ends, because this is going to deprotect the ends of the chromosomes and subject the genetic material to all sorts of instabilities, so the cells shut themselves down and sometimes even commit suicide, rather than have bad telomeres. So they really care about how the integrity of their telomeres is kept. And so telomeres can shorten, as I described, because of this problem of replication. So how is the problem solved? And the problem is solved by this enzyme called telomerase, and we actually made up the name. I'm very proud that we have a word that's now in the Webster's dictionary. We had to make this word up in my lab at Berkeley when we discovered this enzyme, because it was a new enzyme that nobody had seen before. And we found it in an odd place. But I put this picture up because it makes a point. This is a scanning electron micrograph of a single-celled organism. All these little hairy things are called cilia. And this organism swims around. It's in pond scum. And, several would fit on the head of a pin. This is a microscopic organism. And so it's not exactly, a pathogen, a household name, an economically important organism. But it's a great research tool because it has, for reasons that I won't get into, lots and lots of very short linear chromosomes. And even the meanest mathematician will agree, if you have lots and lots of very short linear chromosomes, you'll have lots of ends, right, relative to middles. So that was why I chose it to study telomeres, the ends of chromosomes, and then telomerase, the enzyme that we discovered maintains the ends of chromosomes. So people, here's Carol Greider. She was a graduate student in my lab at Berkeley when we discovered telomerase in a typical Californian pose. And I'm getting a little more technical here, but let me just walk you through it. This is the essence of how this enzyme works. So if you just pay attention to this, you'll know everything you need to know really about telomerase. So now I've blown up the end of a chromosome. So remember I told you about the end of chromosome having repeated sequences, and I've just shown you a few repeats. This repeat happens to be TTGGGG, TTGGGG, and a little bit of it sticks out at the end here. So this would be an end of a chromosome. The whole chromosome would stretch, if we kept it on this same scale, I don't know, many miles away or something. But here we are, and at the very end, this chromosome end sits down on a portion of the enzyme telomerase, which is made up of two parts. It's actually made of multiple parts, but the key ones are a protein and an RNA. What's an RNA? It's like a DNA, but one strand. And so the RNA has bases which are so-called complementary to the DNA here. Now, the rules of polymerizing in DNA world is if you have an A, it's base pairs with a T, and if you have a C, it base pairs with a G. And so polymerization makes base pairs, and what happens is that the DNA gets elongated by the building blocks being put in. So now if this happens over and over and over again, you can elongate the chromosomes as much as you like. And that's the essence of telomerase. And then other cleanup machinery comes in and sort of fills in the other strand by standard things. So that's the essence of telomerase. Okay, what does it matter? Is it just a little luxury that the cells have for our amusement? Or is it actually something that was important? So again, back to tetrahymena, we actually discovered, first of all, in this organism, because that was the place where we'd first discovered telomerase, which is found in all sorts of organisms excepting bacteria, and of course it's found in us. But what we discovered with our initial experimental system way back was that telomerase, as I said, was in tetrahymena, and in fact the telomeres were being maintained because it has plenty of telomerase, and every time the chromosomes get replicated and shortened, telomerase can make up for it. And so it does, and keeps a balance. And so the cells keep multiplying, and so this sort of implies that the cells are dividing and dividing and dividing all the time, continuing to divide, and these cells are effectively immortal. So we have an immortal organism. You just have to feed it and be nice to it, and it'll just grow forever. Okay, so it's truly immortal. Okay, so if you have plenty of telomerase, it's immortal, and the cells can just keep dividing indefinitely. So we played a genetic trick on the cells, and we inactivated their telomerase by a genetic trick. And look what happened. Now the cells divided, and their DNA got shorter and shorter and shorter and shorter and shorter, and they ceased to divide. So effectively, an immortal organism slowly lost its telomeres and became mortal. So that set the stage then for asking questions about telomerase in all sorts of other organisms. Here's an organism that's immortal and has telomerase in it. Normally, take it away and it becomes mortal. So we know we're mortal, so obviously there was a lot of interest in, well, what about in us, in people? So let's just get the idea. Here we have telomerase maintains the ends of chromosomes because it can always replenish the DNA at the ends, the telomeric repeated DNA that are lost. Okay, so a few pictures, because as I told you, I like images. And I was in Berlin not long ago at the Pergamon Museum, which has wonderful works of art from antiquity. And here's this lovely thing, which I'm just showing you, which is a portion actually of an ancient Greek sarcophagus. I'm just showing you the two sides. But here's this lovely kind of thing which reminded me of two chromosomes, right, like here. And there was even this gorgeous creature here, sort of prancing around here, which it sort of reminds me of telomerase, right? So I don't even know what the name of this beautiful prancing creature is, but there it is, visiting the chromosomes. So it's a lovely picture, an image that I think kind of has the spirit of telomeres and telomerase. Okay, so the bottom line is if you disrupt this complex I showed you, the long array of repeated DNA sequences with their enough proteins on them to sheath the ends, then if you knock telomerase down or out, the ends become uncapped. Okay, like the shoelace becomes frayed. Okay, so what about in people? So that was our short description of how we found telomerase and what telomeres are like and why they're important for cells. Every cell in our body needs to have its genetic material, excepting red blood cells. So this is important to know. So the question is, of course, well, what's the activity of telomerase? In an immortal system like tetrahymena, that pond scum organism, it's on all the time. The cells are going on forever. Like Energizer bunnies, they just keep on going. Now, first of all, as we grow, that makes sense. We're doing lots of cell division, and so telomerase has to keep up in all the DNA that's being replicated as all the cells are dividing and the baby is developing, right? And then it remains active in various cells that keep multiplying, and cells that make sense, stem cells. One of the characteristics of stem cells, and a very special stem cell, is our germ cells, right? The cells that give rise to the next generations. they have to be immortal. They have telomerase. It's good, right? And stem cells, they also have to retain the capability to produce new cells pretty much throughout life. They have some telomerase too. That's good. And certain cells, when they're provoked to multiply, activated lymphocytes. What's that mean? When you have a pathogen infection, your immune system cells, including lymphocytes, multiply up and then come in and help attack the invading pathogen. So they have to multiply up, and they rev up their telomerase because they've got to go through a burst of cell multiplications. And then hair follicles. We all care about this, right? Hair follicles have stem cells right at the base. And in order to be able to keep producing hair throughout your life, the hair follicle cells have telomerase in them. So this is all good. Now, people thought that in cells that really didn't multiply very much, that then there wouldn't be telomerase. But what we found actually is that it is still there in many other cells that really, these are just specialized names of different kinds of cells in different tissues, but it was also on in these cells that really are not multiplying. And people thought that it was actually all the way off. And I'm going to show you in the last part of the talk that actually it's on, and the quantities are turning out to be very, very interesting in terms of human disease susceptibility and aging-related diseases. So that's one place in adults where, in fact, telomerase is also on, and we can look at it. We can examine it. And then the other place that telomerase is very active is in cancers. So let's think about cancers. These are cells that have lost control of their cell division. They multiply and multiply and multiply, and they don't listen to stop signals, and they don't listen to where they should be. They just are everywhere, or can go everywhere in the body. And they've lost this ability to stop multiplying. So when you think about it, it would make sense that they had a lot of telomerase. Not only do they have a lot, they've revved it up. It's very, very high levels in cancer cells. So we think of telomerase, same enzyme, same exact enzyme made of the core protein, the core RNA, other factors. We think of telomerase in the human body then as really having two faces from our point of view, from the point of view of our interest in our health or our diseases. Same enzyme in two very different contexts has very different consequences then. So telomerase in the context of cancer cells is bad news. I told you that if you have this pond scum organism and you turn off its telomerase, it goes from immortal to mortal. The same works for cancer cells. And so there's interest in having this as a sort of anti-cancer approach. And there's early development work going on in this context. So in this context of cancer cells, telomerase is cancer-promoting and distinctly bad news. But cancer cells are not normal cells. They have a whole lot of genetic and non-genetic changes that have turned them into the kind of hardened, sociopathic individual cells that they are. They are deaf to all entreaties around them to stop multiplying and stay where they should be. They have gone wild, and they have a lot of other changes. One of the changes is they rev up their telomerase, but telomerase is not the thing that makes them cancerous. It's in that context of all of these other changes that telomerase is such bad news. And so what it's doing, as I said, is here you've got these rogue cells, and now you've got plenty of telomerase, and the cells now just can keep on dividing and dividing and dividing. So it's kind of like an enabler, really. It's enabling these rogue cells to keep doing their bad multiplication. And as I said, that's the context in cancer cells. Okay, so now what about in the context of normal cells? The smiling face will give it all away, okay? Because now in the context of normal cells, cells that are able to listen to all the proper signals and replenish the body throughout life appropriately, in that context, telomerase is turning out to be good news. So the situation in stem cells or germ cells, which will be needed for one generation to produce another, and another, that is clearly, in fact, there's plenty of telomerase, and the telomeres don't ever run down. So, you know, the human race hasn't run its telomeres down. We're still going, and that's because generation after generation, the germ cells the cells that produce the ova and the sperm, those are able to have their telomeres maintained. And stem cells in the body, as one goes through life, also have enough telomerase. It might not be completely enough, but they certainly have enough to keep you going probably through much of life, although we're going to see that that might be limiting. So what about the situation where it's not a balance? If the shortening processes are not balanced by lengthening, what would happen? Let's imagine we have just some telomerase. Okay, so now the cells are dividing, and so it gets a bit shorter, and maybe a bit longer, and a bit shorter, and a bit longer, a bit shorter, but longer, eventually loses the race, right? The balance is off. So over some period, it might be years for all we know, you could imagine eventually the cells would become senescent. They would no longer be able to multiply. And then if you had a bit less, so it's shades of gray here. So what if you had a bit less telomerase, right? Then this whole process might happen, somewhat faster. And if there was none at all, then it would happen even faster still, right? So this is what we think is probably happening in human cells as we go through our stages of adulthood. As we're adults, and then we'll go through our decades of years. So, in humans during life, what happens to telomerase? So I told you all the things we could learn about, or many of the things we could learn about telomeres and their maintenance by telomerase in experimental systems such as the pond scum. But of course, we care about it in humans. So now I'm going to come back, because now I've introduced telomeres and telomerase, so I'm going to come back now to the question that I started with. How do we age? And one facet of this multifaceted process, the one I will show you, it turns out telomeres and telomerase is most relevant to, is the increased susceptibility to diseases. And again, this question of how much is genetic and environmental. I want to digress for a moment for this genetic and environmental, because a lot of statements get made about longevity and how long you'll live and how long your healthy life will be. The term is used, your health span, right? There's your lifespan, but there's also your health span. And that's a term that's more and more meaningful. And so those questions are, being asked in terms of, well, how much is genetic and how much is environmental, life factors, stochastic? We won't talk about accidents or violent, death, but other things in people's lives that are not strictly genetic, and how much is genetic. And so a very interesting study was done, and I'm just going to show you the result here. And it was by the Gavrilova and Gavrilov, and it was published a few years ago. But basically, it was a big database, because they used something called the Gotha Almanac, which has all the records of the genealogy of wealthy families throughout Europe, and they were able to cross-reference and get really good data and believe the data for the lifespan of people in this huge database. And they looked at thousands of daughters, all of whom had grown up, and they were all of European and noble families, so everybody was well off. So you could take away the variable of the fact they wouldn't have enough food or shelter, or being eaten by wolves and things, because they were all well off and socioeconomically also up there, right? So things were sort of equalized in lots of respect. They had lots of data, and that was accurate, because there was a lot of property at stake. So people had very accurate records here. Some of the studies of, centenarians, people who claim to live hundreds of years, it turns out some of these populations that you hear about, some of them, they lied about their ages. So the data have been a little shaky. But this is a good data set, and I'm emphasizing it's a good data set. They also just chose daughters, and so they, as I said, they were all well off, right? And they just chose daughters who had made it, who were 30 years and older. So that got rid of all the variables of childhood mortality and childhood death, and also probably mostly of a lot of childbirth death as well. So it's 30 and older. And they asked the simple question. They said, if we look at the cohorts of daughters born every year and compare them with how long the parents of the daughter lived, what's the relationship? So I'm going to show it to you in a graph that'll look complex, but I want to explain it. So basically, this is something which is saying if it's above this line 0, then that is implying that the daughters are living longer than expected. And so we plotted this against, I'll just show it for the fathers. It was the same as for the mothers, how long the father lived. What was the lifespan of the father? And you can see there was this really straight linear relationship. For the fathers who lived longer than 75, so 75, then your daughter had a much better chance, or had an average sort of chance, but 80, she had a much better chance of living longer than the rest of her comparable cohort, and 90, 95, then the daughter had a much better chance of living correspondingly longer. So there was a real relationship. It was also true for the mothers. I won't show that. And so that so-called residual, this number going up with parental lifespan, that's expected if there's a heritable property to lifespan. So you could say, okay, well, I've answered the question. Yeah, lifespan is heritable, or at least has a good heritable component. And that is certainly true for people who do attain these significant longevities. But look at the rest of, and so genetics is contributing a lot. That's true. But look at the rest of the people. This is actually 70 to 80% of the people. And look at this. It doesn't matter if your father lived to 40 or 65. Frankly, it had very little predictive value. That was most of the people. So that's what you expect if inheritance is unimportant. So if anybody says longevity is inherited, one can say, yes, this is largely true. There's a lot of that for the older people, but it's really washed out or overcome by other quantitatively bigger things for the shorter lifespans of parents, predicting their daughter's lifespan. So anyway, so we can say these other sorts of things, call them environmental, call them stochastic, call them life factors. Other things are quantitatively overwhelming the genetic component. This is not to say there isn't genetic components in here. It's just that they're washed out by these other things that can't have a clear, simple genetic basis. So let's take a common statement from a review. This would be a statement from Atzmann et al. It doesn't matter. I mean, lots of statements like this are made in reviews of aging, which is, this is true, elderly subjects demonstrating exceptional longevity, and there's a lot of genetic component for that, have generally been spared the major age-related diseases, such as cardiovascular disease, diabetes and mellitus, and cancer, which is diseases that are responsible for most deaths in the elderly. Most deaths in the elderly. These are the ones that we care about in practical sense. We love the centenarians, right? But they're a tiny fraction, 1 in a few thousand of the population. They're interesting biologically. But if we want to know what's limiting our lifespan, we'd better know about these ones, right? Because that's the practical reality of what's limiting lifespan. And how much is genetic, how much environmental. And so what I want to tell you about is the work we've been doing in the question of telomeres and telomerase in people with our collaborators, who I'll introduce in a moment, and address this question. Now, already there was some very nice work that came from genetics, and this is a situation where, I've told you about the science and how the science will, science can inform, the bedside, if you will, but equally the bedside can inform the science. And human genetics and the clinic have been incredible instructors of biology. And in this case, it's a rare inherited disease in humans, fortunately very rare, because it's a devastating disease. People die prematurely of progressive bone marrow failure. And so here's their chromosomes, and so here's the pair of chromosomes, one of which would come from the mother, one of which would come from the father. You remember, you get your 2 sets of chromosomes, one from mum, one from dad. And so in these individuals, a genetic accident has happened to one of the telomerase RNA genes. So you get one copy from mom and one from dad. And what happens is that by genetic accidents in these unfortunate individuals in these families, they've inherited a bad copy, just a spontaneous mutation that happened, bad luck. And there are billions of people on the planet. And so this disease, although rare, there's actually now dozens known around the world of families with this. And so that told us something very important, and that was that these individuals, they died and never made it to old age. Their telomeres were shorter. So they've got half the amount of working telomerase gene here. Their immune system becomes exhausted. And in fact, that's the progressive bone marrow failure. Bone marrow is producing your immune system throughout life. They're also cancer prone. And that's an interesting thing because it says that the telomerase is protecting the chromosomes against the cancer-causing instabilities. But it gave us a message. That's the important thing listed here, and that is the full human lifespan. To get into old age, you need both of your telomerase RNA alleles, that means the copy from your mother and your father, to be functional. So it's the quantity of that gene product that matters. So it's not black and white, it's shades of gray, and that matters. So put it this way, you just drop it down to 50%, which doesn't sound like a huge change, but it's a huge consequence. So that's the situation for rare disease, but it said having insufficient telomerase is a bad thing and you can't live your life to a full lifespan and your tissue renewal runs out. And actually other tissues also can't renew properly, and people have fibrosis and things like that because their tissues can't renew and then they get scarring of the liver and lungs. So we now know telomerase, known genetic defects make telomeres short in humans, okay? We know that, okay? What about the more common situation? That's a rare disease. You'd say, well, why does this apply to most of us? So now you could say, well, okay, 50% was low. Yeah, what about 70%, 80%? I mean, you know, what's the deal there? And a very interesting clue came from a study that was published back in 2003. And Richard Cawthon and colleagues did this. And essentially, they just looked at people 60 years and older, and they looked at their white blood cell telomeres. And the white blood cells are your immune system cells out in your bloodstream that people will generously give you for study purposes. Okay? And so they looked at these, and then they followed these individuals for 17 years. And look what happened. People who had shorter telomeres were more likely to die, and particularly more likely to die of heart disease and also from infectious disease. Now, the people who have progressive bone marrow failure with the genetic disease, the reason they die is because they had an immune system that became exhausted, and they die of infections, infectious diseases. So, in fact, this is interesting. So basically, that was it. So shorter telomeres in a population, sort of a statistical way. Okay, now there's plenty of scatter in the data, but this cohort was 140 some people. There was a clear quantitative association with a chance that you would be dead in the subsequent 17 years compared with if you had short telomeres compared with the people who had longer telomeres. So it was predictive. But I want to note right away in a statistical way, okay, because there's scatter in the data. I'll show you some graphs of scatter later, so you just get a sense. There is really scatter here. But the question was, well, what caused what? Had these people always been, you know, fighting these sorts of diseases, and were they kind of sick anyway, and for one reason or other, their telomeres were running down, right? Was it just a byproduct to have short telomeres? Or were the shorter telomeres actually having any causal relationship to this? So we came to one part of the answer through a collaboration. And I want to introduce you the folks, and this is Jolyn in my lab who did the work initially, and my collaborator, Dr. Elissa Eppel at UCSF. She's in the psychiatry department, and she's a clinical psychologist who quantitatively studies effects of chronic psychological stress, measured in various quantitative ways on physiological and health parameters. And then a lot of collaborators here, including Richard Cawthon, I told you about, who'd done that study with the telomere length predicting shorter, worse mortality for the elderly cohort he studied. And then several other collaborators listed here. Okay? So the bottom line was, and I'll show you then the data, but the bottom line is this. Chronic stress, which obviously we perceive through the brain, we're talking about chronic toxic psychological stress, I'll just define it a bit more, you know, that is making the telomeres wear down, and we don't know how, but it certainly is. So let me just tell you how we did this. So now we'll get a bit more sort of detailed, because if you make a broad statement like that, you better back it up. So the first questions we said was, we looked in a cohort that Elissa Eppel had designed of caregiver mothers. And these were caregiver mothers of a child who had a chronic disease of one kind or another, be it autism, be it gut diseases, and various things. They are in a continuing and long-standing stress. As you might imagine, this can be quantified in various ways. They agreed to be participants in Alyssa's study. And they have this kind of stress that has been long documented to have bad effects on health. And I won't go into all the criteria, but essentially it's to do with the situation being out of your control, you're not feeling you have resources for it, it's unpredictable, it's all those things that are chronic situations where you don't have control. By the way, I want to clarify the word stress here. People often get confused. They say, well, you know, we're all under stress. But the kind of stress that many of us professionally are fortunate enough to be able to have is challenge stress. It's challenged, we've chosen it, yes, we feel stressed, but we are not in this sort of chronic situation where the situation was not of our choice and we are not feeling we have control over it or the resources to deal with it or that it has any predictable nature. That's the chronic toxic kind of stress, the kind of stress that I'm under here right now, talking with you here. This is good stress, running a marathon, it's all good stress. Okay, so I just want to distinguish between the two because they're clearly clinically very different. Okay, so what was done in this study was to ask, was the level of perceived stress, and this is a sort of gold standard questionnaire that's been well validated throughout psychological studies. I won't go into the details, but it's called the Cohen's Perceived Stress Test, and you can take it online. And the duration of caregiving, which ranged from 1 to 12 years, was that related to cell aging markers? You know, you can think of aging as the telomeres running down, and that was the criterion we were using here. And these markers we looked at were telomerase activity in the cells, telomere length. So recall, if you've got telomerase that's more active, it's going to keep those cells going for longer, right? And the telomere length will be also, you know, the longer they are probably, in a rough way, the more chance they'll have of being able to divide for more subsequent divisions. So we did this. And so I'll show the results first in this rather dry table. So what we found, first of all, was that perceived stress was associated with shorter telomeres and lower telomerase. The worse your perceived stress, the lower were your telomere lengths, and the lower was your telomerase activity in the white blood cells, the immune cells. Similarly, the number of years of caregiving was also related to these things. And so the lower the telomere length, the longer the number of years people had been under this kind of stress. Now, we corrected for all sorts of things, including age, and these relationships wouldn't go away. Here's a couple graphs. If you look at telomerase in the top quartile of stress, it's lower than in the bottom quartile of stress. It's about half. And telomere length, now it doesn't look very different, but look at the error bars, they're very small. There's very low probability, this is by chance, telomeres in the high stress people were lower than in the low stress people. They were shorter, they'd worn down more. Equivalent to roughly at least 10 years of aging if you compared it with the natural rates that you just see in the population at large. So we found this relationship, and it was pretty interesting because now the question was, you know, was it causal? And in fact, we found that the longer the people had been under this stressor situation, the shorter were their telomeres, as you can see here. Okay, now that's what I mean by scatter. There's a lot of scatter, but the point is that the relationship was real. And so this gave us a directionality. And at last, hooray, we can put one arrowhead into this. We know that from these kinds of things that the chronic stress is making the telomerase lower and the telomere length maintenance worse, right? Because the stress is coming in because the number of years is what is telling you that it's coming in from the outside. Okay. And we know from a lot of science that that'll reduce the ability of the cell with that lower telomerase and shorter telomeres in the long run to keep renewing itself. So stress hormones were, I won't go into all those details here, but the bottom line was this here. And another kind of stress, I'll just show you this one piece of data. We've done it for multiple cohorts now. A sort of dietary sort of restraint, which is where people are constantly trying to go on diets but not succeeding. Actually, they often end up gaining weight. They're unsuccessfully dieting, they're stressing themselves out, and in fact we saw this same relationship with telomeres being shorter. The worse on a scale which is called the Dutch Eating Behaviors Questionnaire scoring, the worse it was. Very striking sort of relationships. Nobody would have guessed this. I certainly would never have guessed this would happen. But this was the thing. if you want to ask what's going on in humans, you ask a human, basically. You can ask a mouse or a rat or a worm or a fly, but you can't get the answer unless you ask a human, right? And so this is what's so interesting, I think, in these studies, because we're really seeing in people what's going on. And so now the question was, so we've got signal input, and we've got this effect where we know very complex physiology will go on, but the telomeres get shorter. Does it matter? So I'll just finish with showing you just a few slides that says, this really matters. Heart disease, something we can all relate to, one of the major killers in developed countries. And so we know the risk factors for heart disease. You know, huge studies of thousands of people in 52 countries have shown this. Okay, and they're all the usual criminals. We know all about these, right? But the one that struck us was a psychological stress. It was the same kind that we had been looking at, and which I just showed you, was associated with less cell maintenance. So we had this, but actually we had all the others as well. In the case of fasting insulin and glucose, if they're high, that's a risk factor for diabetes. All our subjects were healthy. What we found was all of these were related to lower telomerase in the blood cells in our cohort, all of the 6. Let me show you a really interesting thing. Here's the heart function of the people, and we've just divided them in split them down the middle, the half with high telomerase and the half with low telomerase. Very simple-minded. Look what happens. If you look at their resting heart rates, here's the high telomerase people, here's their heart rates, here's their pulse pressure. This is high-frequency heart rate variability. If it's high, it's good, is the bottom line. Here's the result for the sitting, they're resting. For the low telomerase people, look, their heart rate, they're sitting resting, they've been sitting like this for 30 minutes, and their heart rate is still higher, their pulse pressure is still higher, and their high-frequency variability is lower. They're all unhealthy. And then my clinical psychology colleagues do a very demanding thing. They make them go through a lab stress test. 5 minutes of anticipation of a videotaped speech that they have to give, some very unpleasant math, and all the time in a controlled environment, trained personnel stare at them stony-faced. Okay, you get it, right? And look what happens. So their heart rate goes up and their pulse rate goes up, but it's like 2 different species. The low telomerase people have this overreaction, their heart rate really goes pit-a-pat, they can't adjust, their pulse pressure goes up, and the higher telomerase people, they just stay much better. So it's really interesting. Heart functionality of this kind, what we see with the low telomerase group is much more predictive of problems down the line for cardiovascular disease. So basically, and I had to show you smoking in case anybody still smokes on the planet, in California at least. They do on the planet, and I don't know about California. Smokers, non-smokers, low telomerase. The smokers, non-smokers were all over the range, but the smokers were all down there. So I just, if I smoked, I think this would make me stop. Okay, so anyway, so basically just low telomerase in blood cells, just simple blood cells, is associated with 6 major risk factors. Very surprising to us, but all numbers. No causality is implied here, it's just numbers, but we wonder if this isn't going to be an indicator, perhaps, of risks for cardiovascular disease. Now, lots of people have studied telomere length. We are some of the few groups in the world studying telomerase in blood cells, but lots of people have looked at telomere length. And now, over the last few years, all these common diseases of aging are being associated in these multiple studies, and I haven't even done justice to them all yet. All of these are being associated with shorter blood cell telomeres. So this is looking very, very general. And what our studies are suggesting about the stress is that there's some causality, because we know low telomerase will, in fact, decrease telomere length maintenance. And in fact, we know that reduces cells' ability to replenish themselves. We know genetically that if you genetically have less telomerase than normal, that leads to lower length maintenance. And we know that actually causes diseases of aging. That's been shown genetically as well in mice and in human studies. So now we also know from clinical studies, this is not we, this is the world of clinicians has known for a long time, this kind of stress causes disease impact. And so the scientific question for us remains, is it through lower telomerase? We know this is true, we've shown all of this, but is this true? Is that what causes the disease? Or strictly speaking, is it something else? And this is just a sort of a sidebar, a sort of an act going on independent. But I think that there's a lot of reasons to think that it might have at least some really contributory causes. So that's what we are learning, that these are some non-genetic inputs into common diseases of aging. So let me just finish by saying this is our summary. We've learned that in people, poor telomere upkeep, which can be certainly caused by rare genetic causes, but certainly by non-genetic influences, including chronic psychological stress of the bad kind out of control, certainly is associated with, and good reasons to think maybe contributes to, not proven, but certainly some arguments can be made, with disease susceptibility and shorter life. I'll finish with my last little image. So I think of a telomere as this beautiful tree-like structure, right? And so you don't want to have that happen. So I will finish here, and I'll put my acknowledgement slide up, but I believe I take questions for a minute or two. So thank you for your attention.
So to start at the end, where
Yes.
you were talking about how...
Oh, good pun. In the telomerase and telomere field, we always like puns.
Start at the end, right?
We always like puns that include ends. Thank you. You've set the tone. You've set the tone just right.
Excellent. So regarding the impact of stress
Yes.
on the relative health of the
Right.
telomerase,
Yes.
would that tend to lend credence to these theories of, positive thinking curing diseases and that sort of thing? Yeah.
We are currently testing this in some controlled studies now, because there's studies where they've been beautifully designed by colleagues at UCSF, where everything is really equalized, excepting the particular intervention is either some stress reduction or a comparable activity involving all the same face time with health professionals and things, but is not stress reduction. So we want to ask that question in a way that we can get a believable answer, because there's a great deal of anecdotal, fascinating associations out there. And so that's one of the studies that is ongoing to ask that very question, because I think this would be very interesting. If you can modulate this, we clearly know what the bad effect, we know what it's like down the bad end of the scale. What can you do, or can you modulate this? Because in a sort of statistical sense of populations, it's clearly, if you could modulate the telomerase, and if there's this causal nature that we see, certainly from the genetics, suggested from other studies, then that might very well be, in a public health sense, helpful. So I think this is a very good idea. You'd love to pop a magic pill that, tweaks your telomerase up a little bit. There isn't anything, known to be specifically popping telomerase up a little bit, and it's also hard to intervene, pharmacologically in very complex cellular processes, and a lot of clinical history tells us that we sometimes do that at our peril. So these non-medical interventions, if one could do them and show quantitative effects, I think that would be very interesting. No reason not to develop pills, great, but it's not about to happen next week or next year or probably next 5 years maybe. But it's at the moment, I think the bigger picture is you've got this huge fractions of people with these major diseases of aging. They're a big burden for their own lives that are affected, their families, their societies. It's bad. Can you push things, in a better direction? Mm-hmm. I think this is what I've come to be really interested in thinking about. And so we do the telomere telomerase side of it, and our wonderful clinical studies colleagues who design these really well-designed studies, they design these great sort of intervention studies. Shannon, did you have a question?
I'll go on with another one, yes. So one of the first questions that I had come up and that I didn't see you quite address was the implication with cloning. So cloning
is really interesting. Dolly had shorter telomeres than they thought she would have. But the cloned cows, the calves had longer telomeres than expected. So remember, telomerase gets turned on as fetal development happens. And so it actually can, telomerase can elongate telomeres. It doesn't have to keep them just the same. And so it's turning out, it's just different from different species. And so the cloning idea that, the clones would have shorter and shorter telomeres, that missed out the essential fact that there's the existence of telomerase, which in a cell and developmental stage regulated way, it can up the telomere lengths. So it's interesting, Dolly was an N of 1. We don't know what would happen if it was in multiple sheep. I don't know, because it was only one number. But there were multiple calves that were looked at, and when they cloned some calves, and they got longer telomeres. So I think the answer, of course, the question should not be asked in humans, so that's not going to be, but at least in the animal systems where people have looked, it's been both ways.
Interesting.
Yeah. Everybody remembers Dolly.
Are we ready for another go with Mountain View?
Thank you for the talk. I was wondering whether the increased lifespan resulting from reduced caloric intake is also related to telomerase in some way.
Good question. Those two things haven't come together. The telomerase part is relatively recent. The caloric restriction work has been going on for many years, particularly in primate colonies. And no, this is going to be very interesting to ask. We don't know how all that works. But I think it's very interesting because the caloric restriction it puts a very demanding lifestyle on people. And it's not clear how widespread it's going to be for people to be able to adhere to that in a way that would increase their health. It clearly does. They are very healthy. We don't know how these relate, and it's going to be very fascinating to see. Open question. Yes.
Thank you.
Hi, I was with respect to like the mechanisms behind the stress,
right?
Reducing telomerase activity.
What's going on? So one obvious one is stress hormones. So the cells in your body will be bathed by the stress hormones that your brain prompts to get produced, and those physiological pathways have been well studied. So we're actually going to start looking at cells and see whether, if you have more stress hormones bathing a cell, will that damp telomerase down? So if so in fact there are indications from early work that's been done and some published that it will damp telomerase down. So now we can then go inside the cell and say what happens. So I think the physiology has got to be very complex, because we can look at one stress hormone at a time, but the whole body will, of course, produce a cocktail of different changes, and you could probably change them all one at a time. But in a way, I think we sort of step back a little and we say we know a lot of the pathways, but it would be very interesting to know, so we have the one sort of from the brain to the chromosome ends and a lot in between. Now, could you come in and intervene and sort of knock down the stress hormones? Would that help? And again, that really just hasn't been looked at, because it isn't clear that that's the rate limiting quantitative determinant. You see, you could see it happening in the test tube, but you don't know whether that yet will be the one. So at the moment we have from here, the brain, the chromosome ends, a lot in between, and that would be great to do those sorts of studies that you're saying. But again, this is what's emerging, and now to dive deep into it and sort of parse out all its parts will probably be, a lot of interesting things for the future.
Thank you.
Thank you.
I think we have another question. Another question in Mountain View?
Somebody's waving, yes.
Hi, thanks for coming.
Thank you.
I have a question. For telomerase,
Yes.
do we know what causes the upregulation for telomerase to happen in cancer cells?
Yes.
Is there some kind
Yes,
of what's inducing
right.
a higher copy of those?
Cancer cells are real kind of, well, in Australia we'd say magpies. They grab everything they can, right? Cancer cells will use any means they can to upregulate telomerase. Sometimes they increase the gene copy number of, interestingly, it's been seen, the telomerase RNA gene. Sometimes they turn the expression of the gene up, and they can do that in a whole lot of ways. And then there are even more ways that they can turn telomerase up by regulating the amounts of protein made and the ways that the protein is regulated. So we actually know a certain number of pathways, and the answer is that there's a plethora of different ways. It's multi-step. It doesn't just happen, very simply. It's actually a multi-step, quite complex process, and some of it relies on what seems to be some rather accidental things happening in the cancer cells. But then those cancer cells that have had those upregulations of telomerase get an advantage and can elbow out the other cancer cells. So there's no simple thing, there's no simple switch that, you'd love to be able to take the switch and flip it off. That isn't the way it works, because cancer cells have so many different ways that they can upregulate the telomerase. Possibly, if we knew for individual cancers, when we get to the personalized medicine aspect of cancers, as one might hope might happen, maybe you could say, this cell did it this way, and we'll switch, this, and this switch off. But at the moment, the way people have approached it more is to try and just attack the telomerase itself and to try to damp it down. But it's a really interesting question, because the cancer cells are doing it sort of by hook or by crook, essentially, depending on which cancer it is. And it's different, if it's not like all breast cancers do it identically, or all prostate cancers do it identically. It seems to be, again, different pathways get opportunistically used by the cancer cells. Does that answer your question?
Thank you. Yes.
Yes.
Thank you.
So I had one last question.
Yes.
And the one disease that came to my mind that particularly affects people in terms of aging is
Yes.
progeria. Is that?
Well, progeria is these rare genetic diseases where something goes wrong, actually often with DNA maintenance. And people die, tragically young and age at these incredible rates. It's really, the jury is out as to how much of a mimic of natural aging that is. I think there's informative components in there, but like the DNA isn't repaired properly. And one of the things DNA does happen to do when you get older is it gets less well repaired from random acts of damage. And so those processes in certain cases of certain progerias are less well done. And just the aging sort of like thing that happens to these afflicted individuals, you can see results from this inability to repair the DNA properly. But we don't know if that really is true aging. So it's sort of it's very interesting. But as you can see, I think the idea is here, this is not all solved by any means. And I think what you have to do is rely on studies as quantitative as possible in humans, try and sort of get the facts, right? And then we do the ideology later, right? But I think we want to really try and get some facts on the ground. And lots of studies have been done that can be sort of brought into the question. But I think it's a really sort of exciting and still very, very open set of questions.
Very much so. Thank you very much.
Thank you.